I'll wait for 1 2 minutes. For those online, can you hear us? Just click on the sum up. It's okay. Okay. So maybe we can start now. Let's go. Okay. So for this biochemistry session, there will be today 2 talks. One from Olivier, which is online right now, and another one from Laurent Bopp. The talk will be about 45 minutes long, I guess. And there will be time for questions, 10, 15 minutes. Okay, so I'm just introducing Olivier. Olivier is a researcher at IRD, French. It's okay, a French research institute. He works at l'Océan, a French laboratory in Paris. And he is the main developer of the PCS model. So the marine biological model of the 2 ocean platforms, CROCO and NO, and as well as a component of 5 Earth system models. And so he's an oceanographer and marine biochemist, and as well, of course, talks about marine biochemistry modeling. If you're ready, the floor is yours.
Okay. The sound is terrible, so it's quite difficult for me to understand or to follow what you're saying. So anyway, so I'm first. I would say that I'm really pleased to give that talk and to participate to that. I mean, modestly to that training school, and even if I'm doing that talk from online and from France. Anyway, so the objective of that talk, I don't really know how long it should last because I couldn't catch what you said, Renaud, on the duration of the talk. But anyway, let's assume that it's for about 40 minutes because I think this slot is 1 hour long. Am I right when I say that?
Yes, it's perfect.
Okay, so let's say it will last for something like 40 to 45 minutes. And if you have really questions that pressing questions that are essential to understand that talk, don't hesitate to interrupt me and ask the questions you have, especially if it disturbs it prevents you from understanding what I'm saying. I will try to understand the question because, as I said, the sound is really terrible. So we'll see. Okay, so this talk is not about PCS. It's about modeling and biogeochemical modeling, general biogeochemical modeling. So I have no idea of your knowledge in marine biogeochemistry. So it's not a course on marine biogeochemistry. So I assume that you have at least some basic knowledge on marine biogeochemistry. But again, if there are any words or concepts that you don't understand, don't hesitate to interrupt me. So when dealing with marine biogeochemistry, we are dealing with a lot of processes that are presented on this scheme. It seems very complicated, but it's a very simplified picture of marine biogeochemistry. So I won't detail that figure. I won't take 5 or 10 minutes. I could spend at least 5 or 10 minutes on that scheme, so I won't take that time. It's just to say that I admit someone. that there are very, very numerous processes. And so when we are dealing with marine biogeochemical modeling, we have to deal with those processes, at least part of them, and they are of very different nature. So there are some biological processes like photosynthesis, respiration, trophic interactions, behavior. There are some chemical processes like the carbon cycle, and there are some physical processes. And when I talk, so obvious physical processes are transport by the ocean, like mixing, advection, etc. But there are also other physical processes like aggregation of particles, sinking, etc., etc. So it means that there are very, very various processes which need or which don't need to be modeled depending on the objectives we have when studying marine biogeochemistry with a model. So to observe and at least hopefully understand the marine biogeochemistry, there are three different main ways. So the first one is to do observations at sea, and there are very, very different, numerous kinds of observing the ocean, like satellite, cruises, buoys, etc. Excuse me? Gliders and so on. There are experiments in the lab, or not necessarily in the lab, but in what I call controlled environments, and there are modeling. And there are very, very different ways to model the ocean. And so that's the objective of the talk of today, is to give an overview of the way modelers model marine biogeochemistry. Something which is very important is to say that there are not independent means to observe the ocean. So there are tight interactions between the different means to observe or to understand, to study the ocean. And for instance, concerning modeling, of course, you can't do modeling without observations and without experiments. So why modeling the ocean? So that's a very, very general statement about modeling. So it could, it's not specific to marine biogeochemistry. So modeling is a limited, but is also a powerful tool to study the ocean for different reasons. So the first one is that with a model, we can test hypotheses like adding, removing, or change something in the system we are studying, and to look at what are the consequences. And it's, of course, much easier to do that in a model than to do that in the real ocean. It allows also to, it provides also a quantitative dynamical framework. It's useful to assess some unknown rates and parameters, so the ones that are very difficult to observe or to measure either in the lab or in the ocean. It may be used to do prediction and forecasting, but also to study the past ocean, like paleoclimate. For instance, it's used to do a prediction for climate change studies. So a lot of people are connecting. Design, and it may be used also to design an observing system or a campaign. So like to address the question of what is the best sampling strategy. So a brief an overview of the talk I will give. So we will start with biogeochemical and ecosystem modeling, so the different steps to build a model. And then we'll go through an historical perspective, the question of the physiology of the living organisms, the diversity, and some final thoughts on marine biogeochemical modeling. So when doing biogeochemical modeling, the first thing we have to, the ingredient that is in all marine biogeochemical models is the equation for mass conservation in the fluid. And so that's the same that is used in a dynamical model for temperature and salinity, for instance. And so in that equation, there is advection, so the impact of advection on the marine biogeochemical variables, diffusion or non-resolved processes, and the biogeochemical sources and sinks. And that's the objective of that talk, which is to discuss the biogeochemical sources and sinks. So that's the specificity of marine biogeochemistry with respect to other tracers like temperature and salinity. So it means also that when modeling ocean biogeochemistry and ecosystems, we always need some kind of an ocean circulation model. It can be a very simple circulation model like a box model, but we always need that kind a circulation model or a dynamical model behind the ocean biogeochemical modeling. It means also that any biases we have in the simulated ocean dynamics produce biases in marine biogeochemical and ecosystem models. And it's very important to have that in mind when analyzing the output from a biogeochemical model. The biases or the results depend strongly, very strongly, on the way ocean dynamics is simulated by or is represented. And it means also that all the challenges that are related to dynamical modeling are pertinent for ocean biogeochemical modeling, like representing mesoscale, sub-mesoscale, mixing overflows, etc., etc. So there is a first challenge, or there is something very important for marine biogeochemistry, and it's also important for ocean dynamics, but because we need an ocean dynamical modeling, it's also very important for ocean biogeochemistry, that's the resolution of the model, the horizontal and vertical resolution of the model. So generally to improve, it's not the only means to improve the representation of ocean circulation, but one classical way to better represent ocean circulation is to increase the spatial and the vertical resolution. And generally, better modeling of the ecosystem and biogeochemical processes generally requires to increase the complexity of the model, and so the number of processes and prognostic variables. And so we see that in terms of computing cost, there is a trade-off between the two expectation or the two ways to improve things. And just to illustrate that, so when switching from different global configuration in the NEMO ocean platform, so the first Orca 2 is a global configuration at 2 degrees resolution, Orca 05 0.5 degrees resolution, and 025 quarter degree resolution, we increase the cost by a lot. So it means that switching from 2 degrees to a quarter degrees increased by about more than 200 times the computing cost. And for biogeochemistry, increasing the number of variables by a factor of 2 increases the cost by more than a factor of 2, so about 3. So we see here that there is really a trade-off between the two aspects. So I will start with an historical perspective on marine biogeochemistry, which is also a way to introduce the architecture of the biogeochemical models. So marine biogeochemical modeling is not a very old science, but it's not a very, very recent science. So the first biogeochemical model, if we can call that a biogeochemical model, and in fact it was more an ecosystem model, but very, very simple, date back to the middle of the 20th century, so Fleming 1939. And it was a very simple model with only one single variable and not real dynamics, no real dynamics, very simple, the 0D box model. to simulate a bloom in the English Channel, so in the, let's say, the northeast Atlantic Ocean. And so we see on that picture the observation, so that's a dashed line with the dots, and the simulation of the model, the prediction of the model, and we see that the model, of course, is not perfect, but was able to capture the bloom, the timing, and the amplitude of the bloom, and it was a very simple model, so you have the equation there. So a growth term, which is a positive mu p term, and a loss term. Then, about 25 years later, there was the first NPZ type model. So NPZ means that the model had 3 boxes: a nutrient box, a phytoplankton box, and a zooplankton box. And so you will hear a lot when reading, for instance, papers on the marine biogeochemical models, the term NPZ something model. They are all based on that framework. So it means that they are all based on compartments representing the different players of biogeochemistry. And I would say what I would call the modern way or a seminal work on which most biogeochemical models, current biogeochemical models are based, is the Fascia Metal 1990. I don't think I make a mistake. Let's check that I'm right. Yeah, 1990 paper. Sorry. Is the work so by Fascia Metal in 1990 with this NPZD model. So with respect to the previous one, so you have many more boxes, and you have one box which you find in all models currently, which is N. So you know that, P, you know that, Z also, but you have the D letter which corresponds to the detritus. And so the sinking particles. And so all studies at that time were restricted to 0D or quasi-0D framework, so a simple mixed layer model. And they were able, you see some results for the North Atlantic Ocean, they were able to capture some of the aspects, some of the observed variability, seasonal cycle in the North Atlantic. So going to spatially results, so as I said, at that time, they were mostly 0D or quasi-0D and very, very rarely 1D vertical models. And so going to the spatially result model, so the first study was in 1990, so the same time as Fascia, and it was developed in Hamburg in Germany by this guy. You see the picture of the guy, so Ernst Mayr-Heimer. And so it was a global configuration, so a global model with a very, very simple geochemical let's say biogeochemical model in which the bio was very simple. So the bio was only a single equation relating export production, so the production of particles, of sinking particles, to temperature, light, and a nutrient. So there was no P, there was no Z, zooplankton, there was simply an equation, NADOC, I would say, equation, non-empirical equation to predict export production. So it's still used, that kind of model, even if they are more than 30 years old, they are still used in some biogeochemical modeling framework. And so how were they performing those kinds of models? They were extremely cheap, but of course, at that time, it was necessary considering the computing powers that was available at that time. Long-term simulations were feasible, so it was feasible to do paleo, future, or steady-state simulations, and they were doing a decent job at reproducing the large-scale annual mean patterns. So you see an example of that with a section showing phosphate distribution in the Pacific Ocean. On the left, the observations, and on the right, what the model was predicting. So, of course, there are some biases, and you see, for instance, something wrong in the equatorial domain, but the main patterns are reproduced. And what I should say is that current models perform better. So 30 years later, they perform better, but not by a lot. And a large part of the improvements we see for this kind of distributions come from an increase in the horizontal and vertical resolution. So in the better representation of ocean dynamics. So then about 6 years, 3 or 6 years later, there was the implementation of the first NPZD FASHAM-like model in specially reserved configuration. So in 1993, in a regional configuration of the Atlantic Ocean, and in 96 in a global configuration by the same group, so by Meyerheimer and a co-author. And so an example of what was produced. So the fact that now there is an explicit representation of phytoplankton, for instance, allows to use satellite observation, like ocean color observation, to evaluate the model. And so that's an example that is shown here. So you have on the upper plot, on the upper panel, you have the observed, at that time, observed satellite chlorophyll observations. And on the bottom panel, you have what the model was predicting. So the NPZD model used at a global scale. And again, I won't detail the what is simulated and the spatial patterns, but you can see just by eyes that the model is doing a decent job. And so just to say what was the improvement. So I say that with a very simple Meyerheimer's type model, the model was doing already a decent job. And so using more complex model doesn't necessarily improve the large scale patterns. But in fact, using that kind of model, NPZD model, doesn't necessarily improve the large scale patterns, but it improved a lot the seasonal evolution or the seasonal patterns predicted by the model. And so now what do we use? So we use, in fact, the same framework, so NPZD model, but with many more boxes. And so you have an example of that on the left, on the right, sorry. And so you see that it's the same kind of model. So you recognize phytoplankton, zooplankton, nutrients, detritus. But for phytoplankton, you have more than one, for zooplankton, more than one, and so on. So more complex model does not necessarily imply more realistic representation of the system. And so there have been numerous studies showing that. And the drawback of using more complex model, and that's one of the reasons why they are not necessarily more realistic, is that there is a huge set of often badly constrained parameters. So the more boxes, the more processes, the more parameters, and generally it's very difficult to have constraint on those parameters. And so that's another challenge we have in marine, in fact, in modeling, and especially in marine biogeochemical modeling, which is to constrain and to tune the model. And so to set the values of the parameters, the large number of parameters. So there are different ways to do that. So the most common way is the old way, I would say the old way of tuning a model is the hand tuning. So you have a list of parameters, you change the value or you set the values of the parameter, you run the model, you compare that to the observation, and then you have some expert or statistical evaluation of the simulation. You say it's okay, it's not okay. You change a parameter and so on. You rerun the model, you evaluate and so on. And so you can do that 10, 20, 100 times. And it's a very painful process. It can be very, very long, but at least we learn a lot from that process. And so you learn a lot about the way the model is behaving. So you gain some intuitive knowledge. Another way to tune the model is to do some to use some data assimilation approaches. like using linear methods, joint model, etc., microgenetic algorithm, etc., etc. So in theory, it's a nice way to constrain a model, but there are numerous difficulties. And one difficulty, for instance, just to come back to a point, statement or something I said earlier, is to attribute the biases to biogeochemistry or to ocean dynamics. As I said, the prediction or the state simulated by a biogeochemical model is also dependent on the way ocean dynamics is represented. And so how do you know from where the bias is coming? And so using data assimilation can lead, for instance, to overcompensation of errors, which is also the case above for the on-tuning technique. Something that is quite surprising is that this step is often overlooked or not reported in the literature, despite, of course, it is a critical step. And so this is a study published about 8 years ago, showing that, so that did a review of the literature and that investigate if there were in publication sensitivity analysis with the model, so changing the parameters and to see how sensitive it is to the values of the parameters, an evaluation, a validation of the state of the simulation, and a real quantification of the goodness of fit. And you see for the three aspects that you have a large number of studies that don't do that. And even, for instance, for the validation process, if they do a validation, so about 2/3 do a validation, but generally it's not a complete validation. So for instance, the validation of the structure of the model. So do I have the right number of variables? Do I represent the right processes? It's very, very rarely done. And you see also that in more than half of the studies, there is no quantification of the goodness of fit. So generally what people do is that they say, they just compare by eyes to the observation and they say, okay, my model is reasonably okay, which means nothing. So it's not a real quantification, but it's very common. So now we go to the third step. So we saw the architecture of the model, so the basic architecture of the model. Now we switch to the ingredients, so to what is behind each box. And in that case, the living box. And in that case, the case of that presentation, mostly phytoplankton. So to represent physiology and mostly phytoplankton physiology, again, I will adopt an historical perspective, which is relevant there because on the historical axis, we go from very simple framework to more complex framework, the same as what we saw for the structure of the models. So in the 40s, there was the first theory on how to model the physiology of phytoplankton, and it was what we call the mono or the Redfield framework. And so this is a very simple framework. So we say that phytoplankton grows, so the growth of a community or the division of a cell, let's say growth in that case, is controlled by the maximum growth rate and by the external availability in nutrient, in that case, in one nutrient. So N is a nutrient. And so you see in that equation, a very, very common expression that is always very commonly used in biology, which is the Michaelis-Menten term. So you see that it's N over Kn plus N. So the name of Kn, it's called the half saturation constant. That's the value of the nutrient concentration at which growth rate is half of the maximum growth rate. And you see also, looking at that equation, that you have for low nutrient concentration, some kind of a linear increase of growth rate with the nutrient concentration, and then a saturation. So for infinite or for very large and nutrient concentration, growth rate doesn't change anymore. So it's a very, very simple framework. And so one constraint, there are two consequences. The first one is that if you model different nutrients, in fact, they are all related. All nutrients need to be related. So it means that the uptake, they are closely linked and not independent. So it means that the ratios between the uptake of dissolved inorganic phosphorus, dissolved inorganic nitrogen, dissolved inorganic carbon, and so on, they are fixed. So it means that the composition of the population, or the composition, the stoichiometric or the elemental composition of the cell is fixed. That's the first consequence. And the second one is that when in the system we go from nutrients to zero nutrients, growth rate stops instantaneously. And again, when we introduce some nutrients in the system, we go from zero to some nutrients, then cells start to grow immediately. And it's not realistic. Both assumptions, both consequences are not realistic at all. The advantage is that it's very cheap. So it means that to represent phytoplankton, for instance, we need only one tracer, one pool. So in the 60s was developed a more complex framework, which is called Droop or Capron approach. And in that case, instead of representing the cell only, or let's say the total biomass only, we represent some internal stores. So here you have the internal store in carbon, the internal store in nitrogen, so mostly proteins, and the internal store in phosphorus. And so in that case, the system becomes of equations to represent phytoplankton growth is more complex. So there is one equation for the internal store, which depends on the external concentration in nutrients. And there is one equation for phytoplankton growth, which depends on the internal stores. So we see in that case that phytoplankton growth does not depend directly on the external concentration in nutrients. Okay? And it also means that in that case, because we have one equation for each stores, there is no need now to have a fixed composition of the cell. So we can represent independently the evolution of the internal stores in carbon, phosphate, phosphorus, and nitrogen. The second consequence also is that when nutrient falls to zero, then it takes some time for the internal store to fall to zero. And so it means that growth rate doesn't stop, doesn't fall to zero instantaneously. So by doing that, we solve two of the drawbacks of the very simple approach, the drawbacks of, I mentioned, so I won't say that again. Of course, there is a drawback to that approach, is that it's much more expensive. So it means that we need at least 2 up to 5 or even more tracers per pool. So most biogeochemical model belong to these two classes of model. And later, the model was, this approach was made more complex. So instead of representing a general or generic stores, we represent some typical aspects of the cell, so like the chloroplast, the nucleus, etc., etc. And so it means more processes, more variables. And sorry, it doesn't work. So there is a picture missing. But in the years, starting from the years 2000s, we used some approaches, used genomic approaches or genomic techniques. And so a complete metabolic reconstruction of the physiology. meaning that it's very, very, very expensive, because in that case, we need up to 1,000 tracers per pool. And so it's very complex. It's not used yet in any biogeochemical models. There are lots of papers saying that we should use that, but nobody really knows how to use that in biogeochemical models. Okay, I switch that plot. So we come to the last part of the presentation. So we saw the architecture of the models, we saw the physiology of the organisms, but of course, and we saw in the physiology of the organism, we considered only a generic population, so a cell, a generic population. But of course, in the ocean, you have a lot of diversity, like in any ecosystem. And so there is, of course, the issue, the challenge of modeling that biodiversity. So you have different diversity, a way to characterize the biodiversity. So you have the taxonomic biodiversity, you have the functional biodiversity, you have the genetic biodiversity. And so here we talk about functional diversity because we do marine biogeochemistry. So we are not necessarily interested in the character, in the species in itself, but more in their role, their function in marine biogeochemistry. How do they impact marine biogeochemistry? And so we will see a list different types of approach to model that diversity. And so we'll see PFT models, trait-based models, and very quickly, gen-centric models. So the first question is, why does it matter? Does it matter, in fact, to represent functional diversity? Is it important for marine biogeochemistry? So first thing, which is not really saying why, but it's a great, great challenge. And here are two examples where diversity, on the way biodiversity can impact, in fact, marine biogeochemistry. So on the left panel, you have a list of different phytoplankton groups, cyanobacteria, dinoflagellates, diatoms, chlorophytes, and etc. And you have the composition, the elemental composition, so the nitrogen to phosphorus elemental composition of the cell, and you see that you have differences depending between the different groups. For instance, diatoms have a low nitrogen to phosphorus ratio. Cyanobacteria have a high N2P ratio. And of course, if you are interested in the nitrogen cycle and the phosphorus cycle, it means that representing that diversity may be important. And on the right panel, you have a relationship. I mean, it's an idealized relationship between diversity and productivity. And you see that both the mean level of productivity, but also the variance, so the variability of the productivity depends on the diversity. So the first way to model functional diversity is a PFT approach. So what is PFT? So PFT means, and it's illustrated, it has been published or sorry, I don't know how to say that, but the theory has been published, or the theoretical framework has been published by Cohen-Leclerc et al. In 2005. It's a very simple way. So it's based on the NPZD approach. And so you see that you add for different groups or different functions, you simply add new boxes. And so here on that example, you have a box for the diazotroph or nitrogen fixers organisms, you have a box for diatoms, you have a box for coccolithophorids, etc. And so it means that you add one box per function, or one compartment per function. So of course, how to define those boxes? So first, a PFT should be defined when it has a specific biogeochemical ecosystem function. It should be allocated a distinct set of physiological environmental characteristics. If you are not able to distinguish diatoms from phaeocystis, of course, there is no need to split, to separate or to differentiate the two. And a PFT should be important. It should have some importance, at least in a region of the ocean. If it doesn't matter, of course, no need to represent that. The difficulties. So inside or within each box, you have a lot of different species that are lumped into a limited number of boxes. And how do you assign a single set of parameters to those numerous species? The structure of the model is fixed. So it's you can't predict, you can't immerse, the model can't predict the structure of the ecosystem because it's set by the model, and tuning is a pain. I mean, it's a pain. The more variables, the more boxes, as I said, the more boxes you have, the more variables you have. And in that case, you have a lot of boxes, and so you have a lot of variables. So the second approach is a trait-based modeling framework. So, and it's illustrated there. So instead of modeling species or in fact fixed groups, phytoplankton or plankton groups, species in that case are not specifically modeled. What is modeled are the functions by itself. So you don't represent diatoms, you don't represent calcifying organisms, you specify the functions. And on that slide, you have three functions: heterotrophy, so phagotrophy mostly, you have nutrient uptake, and you have light uptake, so phototrophy. So you don't represent any group, you just represent the ability to take up nutrient, the ability to eat prey, and the ability to capture light. And so it means that the structure of the ecosystem or the function of the ecosystem emerges from where you are in that space. So, and in fact, it means that here you define a space and the model predicts where you are in that space. Of course, the difficulties is the number of traits or the kind of traits you want to represent. So here you have 3 traits, but of course you made a choice, and is it the best choice to represent a system? So do you need more traits than those simple 3 traits? Again, also because the model will predict a state where you are in that space, you can't be everywhere at the same time. And you see that on that plot that you need to have some kind of a trade-off. So what if you are here, for instance, in that space, it means that you are good at doing heterotrophy, but you will be bad at doing nutrient uptake and bad at doing light uptake. Okay? And so the question is, if you invest in heterotrophy, it means that you will invest less in nutrients and less in light. And that's the question of the trade-off. So how expensive is it to invest in heterotrophy? How expensive is it to invest in nutrients? And how expensive is it to invest in light? And it's a very, very complicated question. You have also a drawback, but that's the case for all models, that is all traits, everything, all states are achievable everywhere, all the time. Sorry, all the time. And that it may be not easy to follow, but there are some implications for evolution. And it is very expensive, very, very expensive. And you can understand that by understanding that a trait is an axis. A trait, it's a space. And so it means that having, in that case, 3 traits means that you add 3 dimensions to your system. And so if you do 3D modeling, it means that you have the 3 spatial dimensions, and in that case, the 3 trade dimensions. And so you see that the cost increases in a geometric way. So generally, the first trait, or the trait that is all very commonly considered in model, is size. So it's what we call a master trait, because a lot of other processes depend on that trait. So for instance, most or many metabolic rates show some dependency on size. That's what we call allometry, and that's a typical expression of the dependency on size. And you have here an example spanning from bacteria to elephants of respiration as a function of the mass. And you see that you have some kind of a log-log linear relationship in that case. You also have some similar or some allometric relationship between in the trophic interaction. So here that's a relationship between the size of the predator and the size of the prey, and you see that you have in the log space, you have some kind of a non-linear relationship. And you also have some relationship between the abundance and the size. And so you see that the abundance tend to decrease again in a linear way. So it's a power function. So in a linear way in the log-log space, you see a decreasing linear relationship. Most current models belong generally to both formalisms at the same time. So it means that PFT and trait-based approach, they are not orthogonal. So most models mix up the two approaches. And you have here an example of a very, very complex model, which is a Darwin model developed at MIT in the US. And you see here the structure of the model with, on this axis, some kind of a PFT approach. So you have PICO group, CROCO group, coccolithophorus group, nitrogen fixing group, diatom groups. And you have on the other axis, you have traits. So thermal norms, thermal adaptation, and size. And so you see here that you have both PFT and traits. So I will skip the gene-centric approach because it's quite complicated and it's only emerging. So just to say that it's a way to model, to introduce some kind of genomic information into marine biogeochemical model. So in that case, instead of modeling groups, instead of modeling functions, we model genes. So genes are models. And so it's very, very complicated, and it's a very limited approach. Currently, it has been only applied to nitrogen physiology, only nitrogen physiology. It's also very complicated because we don't know for many functions, so for many cycles, we don't know the genes, all of the genes that are implied in the reaction. So there is a limited knowledge that prevents, it's not the only reason why it's not used, but there is a lack of knowledge to really develop that approach. So some final words, I have no idea of what time it is, if I'm late or in advance. I suspect that I'm late, but some final words. So it was a very brief and subjective overview of biogeochemical ecosystem modeling. I hope you were not lost. But if you are, so I had no questions, but I don't know if it means that you understood everything, but there will be space for questions. Many aspects, of course, have been omitted. So I spoke mostly of phytoplankton, for instance. I didn't speak of upper trophic levels like fishes, for instance, but they matter, even in biogeochemical modeling. I didn't speak really of evolution, of niche modeling, of micronutrients, diagenetic benthic processes, etc., etc., etc. Many challenges have not been mentioned, and as I said, I probably don't know all of them. One of these not clearly stated challenges is that, as I showed in my introductory picture, biogeochemistry involves a lot of different processes, and not necessarily only geochemical, biological, etc. processes, but also physical processes, genomic, etc. And so there is a challenge when doing biogeochemical modeling to bring together people having different expertise. And of course, some things that you have to keep in mind in physics, in biogeochemistry, in any type of modeling, models are not the real world. And it's especially true for biogeochemistry or for biology. So models are always imperfect, they are always wrong to some extent, and so they necessarily show some level of deficiency. So you always have to keep that in mind and to remember when looking at model output or when using a model that they will be wrong. They are not the real world. So thank you. I think I'm done. Yes. So thank you very much. And if you have any questions, please, you're welcome. And there is something in the chat. Yes.
Okay, thank you very much. Maybe I would switch on the microphone on my computer for you. If we talk here, is it better like this?
Yeah, it's better.
It's much better. Okay. So, is there any question in the room? And if you have any question, maybe you can get closer from the computer, please. Right. And you online, if you have any question, don't hesitate to raise your hand or to put some questions on the chat. It seems that your talk is perfectly clear.
Okay. Anyway, so I don't know. Oh, there is an echo.
There is an echo?
Yeah, I can hear me, in fact.
Okay.
Oh, but anyway, so 10:30, so
yes.
And if you have any question.
Okay, no, it seems there is no question, so thank you very much, Olivier.
Okay, and so if you have any question later, of course you can ask those questions to Renaud and Christian.
Okay, there is something online? No. Okay. Okay, thank you very much.
There is one question.
There is a question, finally. So, a question from Stanley, more, I guess. So I think you can read it. How do current BGC model deals with benthic pelagic coupling? Good question.
Okay, so can you switch off your mic, Renaud?
Yeah.
Okay. Yes, that's better. So I could spend probably an hour chatting about the way benthic and pelagic coupling is performed. So, but I won't, of course, take too much time. So, in most biogeochemical models that are currently used, especially for the ones that are used in the open ocean, but even those that are used in coastal systems, the benthic system is very crudely, if at all, represented. So you have, in those models, you have two basic ways to model that. Either, no, three basic ways. So the first one is to consider that the bottom of the ocean is a wall. And so everything that reaches the bottom of the ocean is put into the ocean again. So it's not lost to a benthic system. So there is no benthic system, and it's just like a wall. In some other system, it's not really a wall. So you have a wall, you lose some of the materials, and you put back the rest in the. So you have some kind of a burial, permanent burial, and the rest is put back into the ocean. And the third way is to use what we call meta model, like the one developed by Middelburg in the 90s. And so some meta models. So instead of representing the full benthic system, there are some equations saying how much oxygen consumption you have in the benthic system, how much denitrification you have in that system. So that's the three main ways to model benthic. So it means that in most models, there is almost no real representation of the benthic coupling. The other way is to have a full diagenetic model whose complexity is taught by itself, so varies, and you have only very little models that do have a benthic model. So generally, when you do paleo stuff or coastal stuff, you may want to have that, but it's not very common. And just to say that in the PSCS, for instance, there is a benthic model that you can, a full benthic model that you can activate, or there is a meta model. That's all what I can say. The problem with benthic model, just to finish up with that, the problem with a benthic system is that it takes a lot of time. It's very slow and it takes a lot of time to equilibrate. And so that's a real difficulty there is with benthic modeling, is that you have to run the model for in coastal system for 1,000 years, in the open ocean for 100,000 years. And so it's a lot of time, and it's much longer, and it's too long generally for biogeochemistry, too expensive. Okay, I hope I answered your question.
Yes, thank you very much. Okay.
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 So I leave the floor to Laurent. He's online. So Laurent, it's your turn.